Hello friends, welcome you in my YouTube channel. Today in this video I am going to discuss about amino acids and protein. My name is Shirajul Faizi, Assistant Professor, Department of Chemistry, LS College Muzaffarpur. So let us start with the amino acid and proteins. Look at this picture. A lot of protein substance is there. So amino acids are the building blocks of proteins. They contains carboxylic acid group and amino group on the alpha carbon. The carbon adjacent to the CO, which is known as alpha carbon. Because they have both weak base present, they actually exist as a salt. Each amino acid contains a different side groups, means R. So this will vary. This NH2 group and this COH group, and this is alpha carbon and this is alpha hydrogens. The structure of most amino acids. Unlike most organic compound, amino acids are soluble in water, have either high melting point or decompose upon heating. They act both like inorganic material than organic compounds. The molecule is called glycine, where R is equal to CH3. The molecule is called alanine. You see, if R is equal to H, then glycine. If R is equal to CH3, then alanine. So polar amino acid, less polar, and non-polar amino acid. Polar basic amino acid, polar acidic amino acid. So essential amino acid, alanine, histidine, isoleucine, leucine, lysine, etc. required in diets of children, not adults. A zwitter ion has an equal number of NH3 plus and C double bond minus. Group forms when hydrogen from COH group in an amino acid transfers to NH2. So this is glycine, this hydrogen transferred to this nitrogen. So it becomes zwitter ion of glycine. This form is not present. The isoelectric point IP is the pH at which the amino acid has overall zero charge. The isoelectric point of amino acid range from 2.8 to 10.8. Glycine with IP of 6 exists as a positively charged species at pH below 6. So this is the zwitter ion acceptor H plus and positively charged ion. In solutions that are more basic than 6, solutions with greater than pH 6, the NH3 plus loses a proton and amino acid has negative charge. Glycine with IP of 6 has a -1 charge in solution that have the pH below pH. So this is zwitter ion and this negatively charged ion. Aspartic acid and acidic amino acid has IP of 2.8. It is neutral salt at pH 2.8. Forms negative ion with charge -1 -2 at pH value greater than pH 2.8. So look at how aspartic acid, zwitter ion, aspartic acid and different pH. So why is the isoelectric point of amino acid significant? They provide a mean of separating the identifying them. In electrophoresis, an electric current is used to separate mixture of amino acid. The positively charged amino acid move towards the negative electron, and negatively charged amino acid move towards the positive electron. An amino acid at its pi doesn't image. The amino acids are identified as separate band on the filter paper or thin layer plates. So look at here, mixture of amino acid. Now after electrophoresis you have different. Consider a mixture of amino acid, lysine, valine, and aspartic acid at pH 6.0 that is subjected to an electric voltage. So electric voltage which is So you know 2.8, 6.0. So it become 9.7, 6 and 2.8. So positively species pH less than Pi moves toward the negative electrode, and negatively charged species pH greater than Pi moves toward the positive electrode. A species with no net charge does not migrate. Lysine 9.1, aspartame 2.85. So at pH 6, a serpent exists as like this. So all amino acids are chiral except glycine, which has two hydrogen atoms attached to the alpha carbon atom. All amino acids have the same relative orientation of groups in space. The other orientation is essentially not present in living system. It is the L-oriented, and all amino acid possesses, in contrast with sugar, which have the D absolute confusion orientation. So D-glyceraldehyde, L-alanine, and L-lysine. Formation of peptide and amide linkage, glycine, alanine form this. So N terminal this and C terminal from alanine, from glycine and from serine. A dipeptide is named with a while ending with the N terminal amino acid in sequence. The full amino acid name of the phyric carboxyl group COO at the C terminal end. So what is the possible tripeptide form from one each of the leucine, glycine and alanine? What is the difference between leu-gali-ela and ela-gali-leu? So look at the structures, how they are making peptide. So primary structure protein is the particular sequence of amino acid that form the backbone of peptide chain of proteins. You see, this is the primary structure. Non-peptide oxytocin and vasopressin have similar primary structure, differ only in the amino acid at position 3 and 8. This is oxytocin, vasopressin. Insulin was the first protein to have its primary structure determined. It has primary structure of two polypeptide chain linked by disulfide bond, and A chain with 21 amino acid and B chain with 30 amino acid. So primary structure of protein is the particular sequence of amino acid that form backbone of peptide chain of protein. The secondary structure of protein indicate the three-dimensional spatial arrangement of polypeptide chain. Secondary structure and alpha helix. Alpha helix has a coiled shape held in place by hydrogen bond between amide groups and carbonyl group of amino acid along the chain by hydrogen bond between the hydrogen of an NH groups and oxygen of CO of fourth amino acid down the chain. So a beta pelleted sheet is a secondary structure that consists of polypeptide chain arranged side by side. It has hydrogen bond between chain, has R group above and below the sheet, is typical of fibrous proteins such as silk. So triple helix consists of three alpha helix chain woven together, contain large amount of glycine, proline, hydroxyproline, hydroxylysine and other containing OH group for hydrogen bonding. It is found in collagens, connective tissue, skin, tendons and cartilage. Look at this. So the primary structure of protein is the particular sequence of amino acid that form the backbone of peptide chain or protein. The secondary structure of protein indicates the three-dimensional space arrangement of the polypeptide. The tertiary structure of protein gives a specific three-dimensional shape to the polypeptide chain, including the interactions and cross-link between different parts of peptide chain. The tertiary structure is stabilized by hydrophobic and hydrophilic interactions, salt bridge, hydrogen bond, and disulfide. So hydrophobic interaction, hydrophilic interaction, salt bridge, hydrogen bond, disulfide. Hydrophobic interaction between monopolymer groups. Hydrophobic philic The attraction between polar or ionized group and water on the surface of tertiary structure. Salt bridge ionic interactions between the ionized acidic and basic amino acid and so on. So globular protein have the compact spherical shape, carry out synthesis, transport and metabolism in the cell. The proteins such as myoglobin stored and transport oxygen in muscles. Look at this. The primary structure of protein is a particular sequence of amino acid, the form backbone of peptide. Secondary structure of protein indicates the three-dimensional spatial arrangement of polypeptide chain. The tertiary structure of protein gives a specific three-dimensional shape of the polypeptide chain, including interactions and cross-link between different parts of peptide chain. The quaternary structure is the combination of two or more tertiary units. It is stabilized by same interaction found in tertiary structure. Hemoglobin consists of two alpha chain and two beta chain, with heme protein is heme group in each subunit that pick up oxygen for transport in blood to the tissue. So these are the demonstrations: primary, secondary, tertiary, and quaternary. So denaturations involve the disruption of bonds in the secondary, tertiary, and quaternary protein structure. Heat and organic compounds or break part hydrogen bonds and disrupt the hydrophobic interaction. Acid and bases that break H plus bond between the polar R groups and disrupt ionic bond. Heavy metals ion that react with SS bond to form heavy metal sulfide. Agitation such as whipping that stage peptide chain until bond break. So denaturation of proteins occurs when an egg is cooked. The skin is wiped with the alcohol. Heat is used to centralize blood vessels and instruments are sterilized in an autoclave. So classification of subprotein and their function: structural, contractile, transport, storage, hormone, enzyme, protections, and etc. So relative new groups of disease are protein called prions. In the non-infection from the tail is random coil, no regularity in structure. When adjusted, the tail can get folded into beta pileated sheet. It now become an infections agent and has. So friend, hope this video will be helpful to you. If you like this video, then subscribe my